So very good morning, all respected faculty members. So welcome to the third day of EFDP program. And today's session is on, today's lecture is on next generation AI-enabled pharmacophore modeling and 3D QSR for computational drug discovery. So the lecture is delivered by Dr. Perugu Shyam, Associate Professor, Department of Biotechnology, National Institute of Technology, Warangal. So a brief introduction about Dr. Perugu Shyam. So he is an associate professor in Department of Biotechnology, NIT Warangal, with over 15 years of combined experience in teaching, research, and academic administration. Prior to joining NIT Warangal, he served as an assistant professor at JNTU Hyderabad and as a scientist B at Biomedical Information Center, ICMR, National Institute of Nutrition, Hyderabad. So where he worked on computational drug discovery, metabolomics, pathway modeling, next generation sequencing, and development of national nutritional databases. His research focuses on bioinformatics, computational drug discovery, molecular modeling, molecular docking, molecular dynamic simulation, AI and ML in healthcare, genomics, proteomics, and systems biology. He has successfully supervised one PhD scholar and currently guiding two PhD scholars and has mentored over 85 UG and PG research projects. Dr. Shyam has published more than 55 research papers in reputed international journals and authored several book chapters with leading publishers, including Springer Nature and CRC Press. He is also inventor and co-inventor of two patents and contributed to multiple externally funded research projects supported by agencies such as DST, DBT, BIRAC, ICMR, SPARC, and at NIT Warangal. His contributions to academia has been recognized through several prestigious awards, including Best Researcher Award, Associate Fellow of Telangana Academy of Science, National PDF Fellowship, UGC BSR Meritorious Fellowship, and Gate Fellowship. Dr. Shyam has organized numerous national and international faculty development programs, workshops, and conferences in bioinformatics, computational biology, AI, data analytics, and next generation sequencing. He has also held several key academic and administrative positions, including ERP coordinator, NSS program officer, faculty advisor, training and placement officer, and board of studies member at various institutions and universities. He is an active member of several professional societies, including International Society for Computational Biology, Federation of Asian Biotech Association, International Association of Engineers, and Telangana Academy of Sciences. His research continues to focus on developing innovative computational and translational approaches for drug discovery, precision medicine, and biomedical data science. So with this brief introduction, I welcome Dr. Perugu Shyam to take over the session. Sir.
Yeah. Am I audible? Hello.
Yes, sir.
Am I audible?
Yes, sir. Yes, sir.
Okay. Srikanth sir, am I audible?
Yes, sir.
I think sir has a...
Yes, sir.
Yes, sir. Okay. So, thank you, Srikanth, for your wonderful introduction. So, first of all, I would like to thank the organizers for inviting me to interact with the faculty members from across the India. My voice is clear?
Yes, sir.
Chala.
Yes, sir. It is clear, sir.
So, hope you all are the faculty members than the research scholars and PhD students. So, I have a query whether it is a heterogeneous group and homogeneous group. Participants? All
yeah, it is multidisciplinary, sir. It is multidisciplinary.
Multiple disciplines.
Yes.
Okay, the multiple disciplines.
Yeah.
chemistry, biology, microbiology, etc. Pharmacognosy.
No, in pharmaceutical sciences, there are multidisciplinary will be there.
Okay.
Pharmacology,
So all of the
pharmaceutics.
pharmaceutical sciences
Yes,
only.
yes, yes.
Right? Chalo.
I hope. Yeah,
Yeah. Thank you, sir.
welcome.
Sir, can you let me know that my screen is visible?
Yes, sir. Screen is visible, sir.
Okay. So thank you, sir, for responding to my query. So today I'm going to address the next generation artificial intelligence enabled pharmacophore modeling and three-dimensional quantitative activity structure relationships in the computational drug discovery studies. So before going into the today's topic, so what is the generation which we are talking? What is the generation which we are talking? Why we need to say next generation?
Because traditional is going to be upgraded. Traditional,
Any idea?
yeah. Traditional things can be upgraded. Next generation,
Okay, traditional practices to some advanced practices. Is what your answer, sir?
yes, sir.
Okay. So following in a developing of a drugs, but due to the technological changes or advancements in the technology, so there are a lot of the changes that are taken place in developing the drugs. So once upon a time, we used to synthesize, we used to go with the Ayurvedic drugs, making the small formulations in the form of a crude compound. We used to give the medication. Madam, you are speaking to me? Okay. So then subsequently, so we have changed the crude compounds to the extraction of the particular compound from the.
Sir, your screen
Next.
is not visible, sir.
What's that?
That PPT is not visible. Presentation is not.
It is coming. I went to the slide share mode only.
Yes, sir. Now it is visible.
Yes, sir. So, today I'm going to address the next generation AI-enabled pharmacophore modeling and three-dimensional QSR for the computational drug discovery studies. So, where we used to follow the traditional approaches for the designing of a drug and developing of a drug. So, with the in the technology and developments in the high throughput sequencing methods and high throughput assays, then which we are taking the advantage of the technology, and these days the artificial intelligence prediction models are giving the better accuracy results, and they are guiding us to proper way to design a drugs. So when we talk about the pharmacophore, when we talk about the pharmacophore, a pharmacophore model consists of a features. So, what is a pharmacophore and what are the features that has? When you are talking about the pharmacophore, this pharmacophore organized in a three-dimensional pattern. Okay. So, whenever you have a three-dimensional pattern, so we are working with the computationally. When you are working with the computationally, you have a three-dimensional features. These three-dimensional feature represented by the sphere. The sphere is nothing but a particular shape or the variants that are existed or that are surrounded by a particular structure. So when you are talking about the structure, it has a geometrical variations. So these geometrical variations will tell us the radius. This radius determines the tolerance on the deviation of a particular position of a molecule. So whether this carbon 1 to the carbon 2 or carbon 2 to the functional group or one benzene ring to the another benzene ring or benzene ring to the pentane ring or decane ring or etc. So that is the positional deviations that determines the radius. So whenever you have this structure, whenever you have a structure, these structures can be labeled as a single feature or multiple feature or any logical combinations such as and or not. So these are called advanced the Boolean operators and or not. When we used to use the Boolean operators to combine the different interaction patterns, these important patterns within a particular label of a structure. So whether it is the coumarins or quercetins or benjamines or whatever the series of the molecules. So particular pattern of a molecule, you are trying to find out the different interactions. So there are additional features also will be there. So that additional features will be describe the forbidden volume interactions that may represent the receptor boundary interactions. That means the receptor may have the exposed to the surface or it may buried in the structure. So that we have to see in terms of a structural development of a particular molecule. Then pharmacophore modeling. The pharmacophore is a query that comprised in a different features. So what are the features? Every molecule will represent particular property that is called molecular recognize as a motif. So that particular motif consists of a hydrogen bond interactions. So that may acceptors, that may donors, or your molecular interactions are the ionic interactions, cationic interactions, hydrophobic interactions, hydrophilic interactions, and disulfide bonds, salt bridges, and aromatic groups. all these molecular features that represents a particular molecule. Every molecule has some of the donors, some the acceptors, or the hydrophilic bonds, hydrophobic bonds, disulfide bonds, or that molecules may be aromatic groups or aliphatic groups, or that may has the salt bridges and etc. So these particular radius of a sphere, whatever the sphere I am talking, a particular molecule has a particular sphere. So this sphere determines the stretches of the geometric constraints. So whenever we talk about the geometric constraints, these geometric constraints, plane polarization will be there. Plane polarizations, the dipole-dipole moments, and plane polarization moments. And some of the cases we used to see the dihedral angles or the normal or plane, such indicates the molecular geometrical interactions, right? So this is about a pharmacophore. So when you are trying to model a pharmacophore, so you have to look at all these aspects. Okay. Then how do you do? What do you do? And how do you approach when you wanted to develop a drug? So what are the considerations we have to follow? The first scenario is, first you have to identify a receptor. Where you will identify? First, disease is there. The disease associated with a particular gene, particular protein. So the gene is suppressed or protein may be suppressed. So the protein or gene you need to identify from the patient or from the polymorphism. So when you have a particular disease that are there in the society, then identify a receptor or identify the root cause of a problem, then extract the particular material, then whether the DNA, then RNA or protein or some enzyme or some other target. So these targets are maybe any of these. So any of these, the targets will be there. Okay. So whenever the targets are there, so these targets from the across your body, across our body. Then, so whenever the across the body the components are there, so that may be your lungs, that may be your brain, that may be your intestine, that may be your nervous system, that may be your venous system, or etc. The appropriate receptors which we have selected. Okay. So once it is done, then how do you do this? How do you do the particular molecule? So you have to develop a molecule, the appropriate receptor. Okay, you have selected a receptor. Okay, you have selected receptor, then you have identified the target. So from this target, so how many donors are there? How many acceptors are there in a receptor? Then whether ionic bonds are there, cationic bonds are there, or any disulfide bonds are there, and the receptor sphere consists of a what type of the geometrical range, how much radius it has. Okay. Then you have to look into all the studies. So once you study all these aspects, then in terms of a proteomic study, in terms of a genomic study, then you have to look at the particular target. Then in bioinformatics study, computationally, we will screen all these aspects in terms of our computational. So we will identify a biological targets or druggable targets. Then we'll screen lakhs of the molecules with the receptor. We'll come to a consensus. We will come to a conclusion whether the particular ligand will bind to the receptor cavity with these strong interactions with good hydrophobic and hydrophilic interactions, and the ionic interactions, and etc. So the bioinformatics will prove us all these studies. That may be a doctrine, that may be a virtual screening, that may be a network study, that may be molecular dynamics, that may be a molecular simulation studies. Whichever is required, we will perform all these studies. Then, chemist has to synthesize the compounds. So you have a target. The target, how do you fulfill? The target, how do you fulfill? Then you have to design a molecule. So you have to synthesize the molecule based on active site cavity. Based on the active site cavity, you have to design a molecules. Sometimes the synthesis may be possible, synthesis may not be possible, right? So the series of the molecules, series of the compounds they use to synthesize, that are called small molecules. Then they will give it to us, the chemist will give it to the bioinformaticians. They used to perform the molecular doctrine studies and molecular dynamic studies. So that is the small molecule. This is a macromolecule. Small molecule produced by the chemist, and macromolecules will be developed by the biologist. And biologist again conducts the experiments that are called various assays, whether the proteomic studies or genomic studies or the Western blots, Southern blots, and protein assays or enzyme assays and subsequent downregulation studies, upregulation studies, gene expression studies, animal model studies, and etc. We used to conduct. That is called high throughput screening methods are there. So instead of directly checking all these compounds in experiments, we used to check all these compounds in the computationally. So whatever the computationally the compounds will be screened, will be given a good result that will be identified. Then that molecules will be studied in in vitro conditions, right? So this is the integration with the chemistry and biology, that is called chemi-informatics as well as the bioinformatics. So the bioinformaticians use it to work with the integration of these studies. So these days we have a lots of the databases holds lakhs of the molecules. For example, Campbell, for example, Drug Bank, for example, Zinc database, for example, the SNTP database, etc. So each database holds more than 3 lakhs to 5 lakhs small molecules. So these are the synthesized compounds or some of the semi-synthesized compound and some of the natural product library databases also we have. So all these compounds which we take, instead of taking the synthesized compounds which are already available in the globe, which are already available in the repositories, that we will again screen in the computation. Okay, so the bioinformaticians will work vice versa. Then, so whatever the pharmacophore which we are talking, the pharmacophore will work the features that are based on the various molecular libraries. The molecules will be stored in a library that is called the virtual library. So the libraries where you are storing a molecules in the library, normally we used to store the books. But when we talk about the molecular drug discovery studies and bioinformatics studies, we will say the virtual library. So you are saving whatever the molecules which you are synthesizing, whatever the molecules which you are designing, all those molecules are saving in a computational library. Okay, this library represents lakhs of the molecules that are, every molecule has its own conformation, its own shape, its own orientations. That is called the low energy biorelevant conformations will be there. Look at here, the compounds all looks like the same, or any changes are there. For example, this is called methotrexate. Is there any change in the methotrexate as well as the dihydroxyfolate? Participants?
Yes, sir.
So you are there, right? Thank you, madam.
Yes, sir.
So look at
Yes, yes.
the two
Fourth
structures,
position, there is an option.
methotrexate. dihydrofolate is there. So you have a ortho position, para position, and meta positions. R1, number 1, number 2, number 3, number 4, number 5, number 6, number 8 positions are there. So where the changes are there in a methotrexate as well as a dihydrofolate. Both are looks like identical, but one functional group is varying. So where you have a amine group and it is replaced with the oxygen. So that is dihydrofolate. This is methotrexate. So the amine group is here and oxygen group is here. Okay. So when you are taking, when you have a two molecules, this small difference is there. But when you are working with the 2 lakhs molecules and 20 lakhs molecules, how many changes are there? That you have to look at. Understand? Then, so this molecule property is different and this molecule property is different. When you superpose this methotrexate as well as a dihydrofolate, most of the cases where this amine group, and this amine group, NH2 group and NH group is properly superimposed, and everything will be superimposed, where only NH2 and this NH2 group and the O group will not be superimposed because the conformational changes will be there. But similarly, when you have other than the one more benzene ring comes, then it will be there, right? So look at the structure here. The hydrogen bonding patterns will be differs. So when you are superposing the various n number of the pharmacophore, only basic scaffold may aligns, but your R1, R2, R3, R4, Rn functional groups may not be superposed properly because the side chain will be vary from the one molecule to the another molecules. Am I right? Is it clear?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes.
Okay, sir. Chalo. Look at here. Tomorrow, I have a molecules, but I have to work with any one receptor. So here, my case is EGFR receptor, epidermal growth factor receptor. So that receptor I have to inhibit with some chemical molecules. So which chemical molecule will have a stronger affinity, or which chemical molecule or small molecule will come and inhibit my EGFR receptor with strong affinity or with higher inhibitory values that are called IC50 values. Look at here. This is a molecule. Give the in IC50 is of 10.49 micrometer. Then here the 3.54 micromoles, and here the 0.57 micromole, and here 25.12 micromole and 25.12. So when you look at the log p values, the log p values is the 4.13 and log p values of the 4.96 and 3.91 and 3.57 and 2.3. Then when you look at all these superimposed molecules, then one molecule is designed as a 11 plus C. So this molecule in combination of all the small molecules, we were finalized a one molecule that is called this molecule. This molecule will be fulfilling all the molecular properties to one molecule with the stronger affinity. So what are the steps we need to follow? What are the steps we need to follow when you are trying to develop a pharmacophore? The first thing is, what are the molecules we have to take? What type of the molecules? What type of the group? What group of the molecules we have to select? Whether it is a aromatic, aliphatic, or any salt bridges, or any the gelates, epigelates, or the quercetins, or coumarins, or benjamines, or amines, and etc. groups we have to check. So we are looking at all the aspects of the molecules. So that is first case is information input. Then you are taking the molecule. So let's assume a Tamil Nadu and Karnataka, Andhra Pradesh, Telangana, Kerala, and Punjab. When you look at our body patterns are same or slightly change. When you look at our heights, we all are 6 feet height or 5 feet height or any differences. Then whether all our food habitats are same, slightly differs, right? So that means, just this is an example. When you are taking the molecule, all molecule will not have same size, same shape, same function. Molecules do not have the same shape, same structure, same conformation, same R groups. Then you have to search the conformational search to find out the similar molecules. That is what the conformational search you have to do. When you're working with the molecules, large molecules will undergo lots of the conformational changes, right? Then you have to represent the molecules. This is the structural representation. So you have to make the molecules into a group and you have to make the particular representation. Then every molecule have a particular pattern, right? So particular pattern you have to identify. You need to make a cluster. Then you need to give the, oh, this 100 molecules here has a particular pattern. This 20 molecule is another pattern, and this 30 molecule is another pattern, and this 2000 molecule is another pattern. Then once you have made a patterns, identified a patterns, then you have to give the score. That is called scoring. So each molecular property and you will give the score. So how do you do that? Based on the hydrogen bond donors, hydrogen bond acceptors, hydrophilic property, aromatic property, ionic property, and cationic property. Then shape and geometrical configurations and the properties of a constituents, descriptors. So all these aspects we have to look at. These are the steps which we need to follow. So these molecules you have, the ligand receptor. Ligand receptor means you have a small molecule, then you have a macromolecule. So when they bond together, it will make a complex. So that is the directly you are performing a pharmacophore modeling to extract the feature. So that is called defining a pharmacophore features. But the receptor has particular active site cavity. So that active site cavity we have to see, that is the indirect method. Then identify the common features that are capable to bind the active site cavity. Then propose the drug candidates. And we have to score and rank the pharmacophore candidates. For that, you need to synthesize, you need to hypothesize. So which configuration way the molecule will bind? What type of the ions or molecules, atoms to be placed? Then you have to do it, leave one out, or check the error prone rates. Then you have to decide the molecules. So another approach is called, you need to search the databases, then find out the molecules which are similar, which are identical to the molecules. Whatever the molecules which you are identified, then you need to check. You need to check the lead molecules from the group of the molecules. Let us assume you have 1,000 molecules. The all 1,000 molecules does not have the same property. And all molecules, the 1,000 molecules will not act as a drug. 2,000, the 10 molecules or 5 molecules or 2 molecules will act as a leads. The lead selection to be done from the hits. To identify that, you need to conduct some of the experimental studies that are called high-throughput screenings and high-throughput assays you need to conduct, then optimize the molecules. So, right, like this we used to do. So, this is the simple layman understanding. I have these many molecules. Okay, these many molecules. How many molecules are here? These are just I have represented as a shape. Assume that these are your ligands. Assume that these are your drugs. Okay. How many are here?
9.
So 9 molecules are there. Look at here, madam. These are the my ligands. This is my receptor. Okay. This receptor has the active site. Whenever I take these 9 ligands, whether it will come and bind to my receptor active site cavity?
No.
Then what I have to do?
We have to change the structure of the molecule.
Okay. So these many compounds you have, but how do you design?
So from the 9, anyone we can take.
Assembly, I am in my cabin. Okay. That you no, that you need to map it actually. I will come. I'm in a talk. Yeah. Okay, no problem. Let it be Narasimha, then next time you can do it. No problem, we will do it. Can you inform Narasimha sir once? No, mine is not required. Yeah, in my login it will not be there. Either you, no, either you or either Narasimha sir. That's all. Nothing to worry. We will resolve it. Okay. Just I'm in a talk. I will call you. Okay. Madam. Sorry, madam.
It's okay.
What we have to do, madam? Similarly, you look at here, madam. How many keys I have? I have a bunch
9
of
keys,
keys.
9 ligands.
Yeah. So here 9 ligands we have. So all these 9 ligands will bind to my receptor?
No.
Yes, absolutely. How you are telling?
So depending upon the active site, we need to select the ligand. So which is going to fit to the receptor?
Here you have a proper
active site.
idea about the shape, right?
Yes, sir. Yes.
Based on the shape you have an idea about the active site shape.
Yes. Yes, sir.
Okay. So you forgot about the geometry, you forgot about the topology, you forget about the topological nature, you forgot about the radius, you forgot about the configuration. Based on the shape, you are saying that these 9 ligands will not fit into the receptor. Then again,
Yes,
you have
sir.
to design, right?
Yes, sir.
To fit the molecule into the active site.
Yes.
So here I am shown only 9 molecules. Tomorrow it may be 90,000, 9 lakhs, or 90 lakhs molecules. Understand the background.
Yes, sir. Yes.
So we are not supposed to take a one molecule and to do the docking. That is a wrong scenario everyone used to do. You have to
Yes.
check properly whether it is active site is blocked by the ligand or not. Confirmational search will be there.
Yes, sir.
Always when the ligand come and binds to the receptor, conformational changes will occur. Conformational changes that may take place in the receptor side or ligand side.
Yes.
Okay. Then, so that conformation again we have to study. We have to measure. Then, look at here. If you understand that, if you understand here clearly, then this is the scenario. This is the scenario. So I have a receptor here. This is called my receptor active site cavity. Now I have to design. I have to design a pharmacophore.
Okay.
How do we design? What? See, here NH is there, O is there, here the aromatic ring is there, some aliphatic ring is there. How do you design a ligand for my active site, my binding site?
We need to go for different
90%,
conformations.
90% of the peoples will not do these studies and may not aware also. It's not blaming, they may not aware this. So what do we do? How do we do?
Sir, as you
Please
said,
try to respond, whatever it is.
sir, as you said, depending upon the receptor active site, we need to go for different conformational changes. So based on that, we need to fit into the active site, or we need to change.
Yes, absolutely, madam. Absolutely right. But how do you design? Then you design a drug, no? You design a ligand to fit into the active site cavity. I'm showing my active site. I have
Yes,
shown
sir.
the counter ions also. Side chain molecules also I have shown.
Yes.
Any other participants can reflect?
Fragments we can use now. Fragment based design.
Okay, sir, fragment based. But whatever the fragments you are taking, no, finally you have to bring the one particular compound, right?
But depending upon the active site, whatever we are selecting the protein, based upon that, we will be selecting the molecule.
Absolutely, madam, you are right. And prior to you, one more madam said that is right. But I clearly shown the active site cavity, what type of the molecule you will design. See, look at here. Here you said this nine molecules will not fit into the receptor. This molecule will fit, and you are designing, right, for this shape.
Yes.
Similarly, I have this active site. Then you have to design a ligand.
So we can design the ligand based on how it can make interaction with the NH, C double bond.
Very good. Very
Bonding
good. Right,
interaction.
man. Right. See, look at here. Here, what is the molecule we have? NH. The receptor containing the amino acid as the NH is a side group, side chain. Then what type of the interaction should be there here? So O minus should be there. O minus and this NH plus make a interaction. Understand? Then here you have an oxygen group. Then what type of the counter ion should be there in the ligand?
Should be positive.
Hydrogen.
Hydrogen should be there. Then, okay, you made oxygen here, you made hydrogen here. in the ligand side, but where they has to sit. Understand? So this space has to fulfill, then you have to design a molecule, that is whether it is a pentane ring, benzene ring, or terpene ring, or heptane ring, or hexane. You have to look at all these aspects. Understand?
Yes.
Yes, sir.
Yes, sir.
See, I am taking time to make you to understand. This is the background, sir, madam. Understand this concept. Or you, probably you all knows. If you all knows very well, you please let me explain. I am assuming that you're not aware. If you are aware, just you let me know. So in this Malik figure, I have shown the OH and NH. Then they make a interaction. Then where this oxygen group has to there, then I am keeping some pentane ring. Right? Whether the pentane ring is sufficient or not, then pentane ring you kept. What type of the other molecule will be there? Here, lot of the space will be there. Then what type of the compound should be kept? So benzene ring, I assume that benzene ring, but there is a slanting, the shape is there. Then, so this shape, what I have to do it? Then here, the two benzene rings I'm adding. Then, whether that is possible or not, then I am keeping two pentane rings, one pentane ring here, one pentane ring here. Then I am doing two benzene rings here and here, one more, two benzene rings and another benzene rings. Then you have to check whether it is binding or not. Whether it is properly fitting to the receptor or not, you need to check. So like this, here I have shown only 5 sets of the molecules. But tomorrow you have to make 5,000 sets, 5 lakh sets. Understand?
Yes, sir.
If I am wrong, please let me know.
No, sir, clear.
Chalo, this is the way the pharmacophore modeling can be done. Or you can look at the another aspect. You wanted to define a pharmacophore. Yes, this is called receptor, then active site cavity, then you are making the side chains, then you are searching the database against the ligands, that is the DB search, then you have taken a radius and side chains of the molecules, then trying to find out a molecules, then based on this, and based on this, and based on this, you found here some of the molecules from the database. Understand? So this is the database search for the pharmacophore. So you first understand the what type of the molecules to be useful, or that molecules are benefit to the receptor active site cavity. That way you can keep on search the database and try to find the better molecules. So look at here, building the molecules and the binding site. So this, the molecule radius you have to measure, and you can fit into the receptor cavity and evaluate there the disposition space. Okay, clear? So these are called hydrogen bond interactions, hydrogen bond interactions, hydrogen bond interactions, and this the dipole moments. All this we can evaluate the interatomic interactions, that are the receptor ligand interactions when they bond together. Right? Clear pharmacophore? Chalo.
Yes, sir.
Then we have the molecular descriptors. Whatever the molecules I have shown here, madam, whatever the molecules I have shown, here 6 molecules or 5 molecules, here is a 9 molecules, assumes that these 9 and these 5 are molecules. These are, in my terminology, they are quantitative, right? It's a quantity of molecules. What is the quantity? It's a 9 molecules is a quantity, 5 molecules is a quantity. So when I'm talking about my drug discovery, it's a quantitative. What are that 9? What are that 5 molecules? It has the structures. It has structures. Every structure has some activity. Then every structure has a relationship. That is what the quantitative structure activity relationship. So when we are working in the drug design and drug discovery studies, the quantitative structure activity relationships will be the modeling on a numerical representations, that is regression, correlations, principal component analysis, t-test, and etc. are there. When you wanted to represent that, you require numerical values. So whatever the molecules which you have, you are representing in a numerical form. So numerical representation of a chemical structures. So that they will translate into a physical, geometric, electronic, and topological molecular features into a data that will be represented in my computer algorithms or computer programs. These computer algorithms, computer programs can predict the biological activity or the molecular toxicity. Right? So the quantitative structure activity relationship is a modeling technique that predicts the biological activity based on chemical structure or enabling the prioritization of a compounds for synthesis, testing, and lead optimization in a drug discovery and development approaches. So whatever the drug discovery studies which we have, that is based on the structures, based on the molecule, based on the data that is available with us. It is the data-driven approach that attempts to correlate the chemical structures in the form of a molecular descriptors. So what are the molecular descriptors? The molecular descriptors will be different types of the molecular descriptors are there. So these descriptors, the basic descriptors are topological descriptors, physiochemical descriptors, electrolyte descriptors, chemical fingerprint descriptors, and geometrical descriptors, and QSR descriptors, QSPR descriptors. So these are the basic descriptors. There are the 200 types of the descriptors are there. I'm not talking about all those 200 descriptors due to the time constraint. Then we have only 6 descriptors, basic 6 descriptors. These 6 descriptors will have all ligand molecules. So when we have the machine learning models and the predictive models, these models will follow the tailored drug design approach, either structure-based activity algorithms and integration with the omics data. Then we have a risk mitigation and identifying ineffective compounds in early phase development. So look at here, we don't know the molecular properties of a molecule initially. But when you are identify a patterns, when you are grouped into molecules based on the shape, based on the size, based on the property, based on the function, then that is called known activity. So once you know the activity of a particular molecule, then you can perform a QSR studies where you have an unknown activity, then you are finding some activity. Where you don't know the particular molecular property, where you don't know the particular function of the molecule, then you have identified their function and property and descriptors, then you are predicting a function by using the QSR. So that will give you, that will use the regression value, that will use the correlation value, that will use the leave one out approach and this square fitting, etc. So that will give you the parameters, right? Then, so the constitutional descriptors, this is called zero dimensions, that are the primary construct. The constitutional descriptors, when you talk about the constitution in terms of a general aspect, we belongs to some X constitution. In the X constitutions, in terms of a political elections, all same type of the community peoples will living together. Whether only humans are living in the particular area, geographical area, no. So when we say the constitution, maybe X community, Y community, Z community population will be there. Our neighbor may be X community. Our other neighbor may be Y religion, and other neighbor will be Z community. So religions will be different, communities will be different, and our color appearances also differs, and habitats also maybe differs. Similarly, along with the humans, along with the male and female, we also have the animals, microorganisms, and plants, and some of the other organisms. So these all are there in the constitution. So when you talk about the constitutional properties of a molecule, these molecules have molecular formula, molecular weight, molecular number, then number of the rotative bonds, number of the hydrogen atoms, number of the carbon atoms, number of the other atoms, then whether it is aromatic ring, whether it is aliphatic ring, whether it is a etc., all the types of the properties we need to check. That is called constitutional property. Then topological property, it's a one dimension. The zero dimension is a constitutional property. One dimension is the information about the molecular fragments and how the atoms are connected, including the structure and counts and branching and connectivity indices. So topological properties. The topological properties are molecular fragments. Atoms will be there. That atom is connecting to the which atom? Then including substructure, there are the substructures are there, no? Aromatic ring with the pentane ring with the alkane ring, and furan ring, nitrobenzene ring, and etc. are there. That is a structure count. And branching units, various branches will be there in the molecules, and the connective indices that are called topological descriptors. Then geometrical descriptors, spatial orientation. The spatial orientation of a molecules that are derived from a three-dimensional molecular conformations such as molecular volume, surface area, polar surface area, buried surface area, and shape of the parameters. This is about the geometrical. Then quantum chemical, and that means electronic properties. So van der Waal interactions, electrostatic interactions, and dipole-dipole movements, and partial atomic interactions, partial atomic charges, positive and negative, and orbital energies that are called HOMO and LUMO. So these are the chemical, the properties. Then physiochemical properties. These are the basic physiochemical properties. Every molecule has the micro and macroscopic predicted or experimental the properties like octanol water partition coefficient or molar refractivity, and these molecules ability to cross the biological membranes. So this is called permeability, blood-brain barriers we used to say. So all these will help us the physiochemical properties. So the hydrophobic descriptors will be there. That is called log P, octanol water partition coefficient. So that will predict the lipophilicity nature. And the hence substituent constant, that is called P pi, or pi quantifies the hydrophobic contribution of a particular spatial chemical substituent relative to the hydrogen atoms. So that is about the hydrophobic, and the electronic descriptors are the Hammett constants. This is Hammett constants where the electrons are withdrawing or electrons are donating. So that will be measured in a reaction center. And HOMO-LUMO energies represents the highest occupied or lowest unoccupied the molecular orbital energies in terms of oxidation reduction reactions. Dipole-dipole moments will measures the polarity and charge distribution of the particular molecules. So steric ring descriptors, molecule undergo lots of the steric conformations. So when the molecule has the R group, the molecule will go conformational changes, either this direction to this direction or this direction to this direction, based on the either receptor cavity, either ligand nature, the molecule undergoes the steric configuration changes. That is called, it measures the physical bulk and spatial size of a substituent, that it determines how it hinders the receptor binding. So molar volume or van der Waals volume that estimates the three-dimensional space of a particular occupied the entire volume of a specific groups. Then we have a topological and constitutional descriptors, the molecular weight, Lipinski rule of 5. You all knows the Lipinski rule of 5. And connectivity indices, this encodes the degree of branching or the atomic connectivity of a molecular graphs. So then you can have a particular molecular property predictions by using the QSR, right? So this is the experimental properties of a molecule. When you are talking about the surface area, so the area may be a EM or AFM will be calculated. And when you have a molecule, when you are talking about the EM and AFM, so that will be measured in terms of a diameter. That is called how much flow is there and how much DLS is there. So when you are measuring the flow, then volume will be there. So that is the sedimentation rate will be there. Then you have a hydrophobicity. The hydrophobicity will be liquid extraction, chromatography methods are there. Then aggregation will be there. So DLS, AFMS, and ESEM will be there. Then we have experimental compositions, ICP, ICPMS, ICP-OES, and TEM and EDOX, and the EFS and ICPMS studies. And crystal structure will be predicted by the X-ray and X-ray diffraction and TEM and XRD studies. If you have a surface study, then you can have a zeta potential, the size variations, then electrophoretic mobility studies, and hydrodynamic properties can be measured with the DLS studies or the flow FE studies, and equivalent pore size diameter, that is called particle motion and particle filtration properties will be there. So when you are performing all these molecules, or you may use the two-dimensional NMR or three-dimensional NMR, so that also will predict the experimental properties of a particular molecule. Then you have a feature selection. So you can use the filters and wrappers and embedded, and you can select the molecules. Yeah. Have you ever seen this kind of the set of the molecules, participants? Have you ever seen this
Yes,
kind of set of the
literature, in the
molecules?
literature, sir.
Madam,
In the literature, sir.
the literature you have seen, right? So how many are there? So this is normally, these are normally molecules that are synthesized. These are the molecules are synthesized and functional groups are varying, R groups are varying, and some way the scaffold or mother structures also vary. This is one series. When we talk about the series, it's a one series, but the researchers use it to synthesize a number of the series. So each series may have 100 molecules, 200 molecules, or 10 molecules. So n number of the series of the molecules will be designed from a one research group. When you look at the globally, lakhs of the molecules will be synthesized. So once it is synthesized, somewhere they used to stop, right? So this is quantity. This is called quantity. All these molecules has the structure. Every molecule has activity. We are trying to find a common relationship. Okay, so that is the QSR. So here the constitutional or electrostatic or other properties, these colors are representing, no? These are called the properties or descriptors: electrostatic, constitutional, topological, ionic, and the hydrophilic descriptors. So when you have a molecules, you align the molecule. That means superpose the molecules. Molecular alignment is nothing but a superposition of a molecule. Why we need to do the superposition? Trying to identify the scaffold molecule. That means the mother scaffold is nothing but at least this is the scaffold, mother structure. Then you have R1, R2, R3, R4, R5. In some cases, the mother scaffold also will be vary from the one molecule, one set to the one series to the another series. Okay, we are trying to identify a scaffold. Again, so you perform a virtual screen, take a series of the molecules, perform the virtual screening with the molecular receptor, then identify the molecules where you have the strongest affinity or efficacy or better hydrogen bond interactions and etc. Then perform a molecular dynamic studies. They evaluate the molecules in terms of a root mean square deviation, root mean square fluctuation, and identify their interactions in terms of hydrogen bonds, energy, and dipole-dipole moments, radius of gyrations, and disulfide bonds, and etc. Other properties we can able to predict. Then, so you have these all the properties you have. Then these properties you can have a van der Waal interactions and electrostatic interactions and other property interactions which you have. So you can try to identify all these type of the scenarios in terms of the molecules, right? Then, so you have a library, the laboratory collections. These laboratory collections and literature, so the molecules will be collected from the literature survey and literature records and papers. And you can conduct the computational QSR modeling. And you can do the large chemical libraries where you have molecules that are deposited in the library. Then you can screen the molecules based on the molecular docking, the performing the virtual screening. Then you can conduct the experimental studies in terms of in vitro assays then you can, whatever the in vitro molecules will give you a better results, those molecules you can check against the animals. That is called in vivo studies. When the in vivo studies gives a good results, that is well and good. If not good, then again you can go and search the molecules. Again, you come back and synthesize the molecule from the laboratory. So this process will be continuous in terms of the studies. So this is the overall scenario with the target selection and data set design, data curation and data processing and pre-processing, then descriptor calculations. So there are so many tools and packages are available today. Then we have a feature selections or feature filtering and selection, and model development and benchmarking, and internal validation and external validation, and randomization and applicability, and large scale selection and large scale predictions. Okay, there. So what is the next generation AI enabled pharmacophore modeling in terms of a QSR? So this, look at here, you have a artificial intelligence. How much time I have, organizers? Okay. Is it clear up to here?
Sir, up to 30 minutes, sir.
Okay, sir. So participant, is it clear? Or any doubt?
Clear, sir.
Clear only.
Okay. Thank you, sir. Chalo. So here, that is the basic scenario which we used to do. So today, which we have a artificial intelligence and machine learning methods. So this first we are trying to identify the targets, that is the biological target. That may be a genome, that may be a protein, that may be a gene, that may be a protein of your target. Then why we need to use the artificial intelligence? Artificial intelligence is a faster and cheaper and smarter in the drug discovery studies. So then we are computationally identifying the hit molecules by using the artificial intelligence based on the predictability property. Then based on the predictability property, based on the molecular properties, we are predicting using ADMET properties. These are the physiochemical properties and pharmacodynamic properties. Then biological activity. Then we are finding out the lead molecules. We are optimizing and minimizing the molecules from the large set of the molecules and the library of the molecules. So based on the prediction of a properties of a ADMET. So these are the physiochemical properties also we will look into that. So then once you identify that by using the generalized neural networks or deep learning models and other parameters, then we used to use the predictional property based on the toxicity or based on the target effects. Then preclinical studies will be there. The preclinical studies we used to conduct in terms of in vitro studies as well as the in vivo studies. Then once you validate in vitro and in vivo, then we can go for a clinical trials, and healthy volunteers will be recruited, and that compounds will be checked against the patients. This is, I may not say patient, it is a human volunteer. Then once it is given a proper result, and we'll go for a regulatory approach, right? So if this whole scenario works well, then it will go for a approval. If something goes wrong, then you have to come back here, right? So it is an economical burden and time and laborious and expensive, and all these aspects will be there. So that is the reason it's a high attrition will be there, and long timelines will be there, and high cost. When you really design a drug, normally it takes 12 to 15 years of a time. But these days, by taking the artificial intelligence advancements, we are designing a drug, and we are developing a drug in 4 or 5 years. Then, so here the artificial intelligence, the generative models and de novo drug design is there. So these generative models, how it works, we have a model, we have a deep generative model. So this generative model, how we have designed, we have a compound. This compound, which are synthesized, which are developed in my laboratory, that are called novel molecules. Okay. Then whatever the molecule which I have developed, that molecule has some property. That pharmacological property has. Then this molecule may has effectiveness against my EGFR receptor, my RAS or my BRAF, and this molecule will have a solubility, right? So solubility nature will be there. So that we will check. Okay. So we have to design a molecule in terms of the solubility, in terms of effective working or effectiveness and physicochemical properties. Then we have a molecular foundations. What are the foundations? First, you need to undergo with the literature and study the statistics and case statistics and biomedical and chemical or molecular data sources in a various representations. So sometimes data may be there in a electronic form, sometimes data is there in a documentary form, sometimes the data is available in your peripheral devices, data may be available in a statistical form. Then all these models you have to collect. Then you have to train the models. So data I am collecting from a Zinc database, Federal libraries, or GCSD and TCA, or Geo database, or etc. databases whenever you are taking. So heterogeneous data will be there. You need to train the data. So you need to filter out, you need to classify the data, then you need to pre-train the model and train the model, divide the data into a training set and test set, and make a model. Then you have to adapt the parameters. So how do you design the drug and how do you develop a drug? So based on the disease, you are developing a molecule in the laboratory, then you are reaching to the patient. Okay, so that is you conducting a various studies, literature review, and identifying a molecule and studying their molecular properties, molecular representations, and synthesizing the molecule, conducting the experiments, and clinically trial, preclinical and clinical studies. Then you are bringing the molecule into the market, right? So then. So you have a AI workflow. So whatever the molecule you are taking, first identify the molecular properties. So that properties are whether this molecule can able to bind, that is predicting the ligand binding nature. And simulating the molecular behavior, how the molecule behaving in a time intervals. Time intervals, how the molecule is behaving. Then calculate the free energy, delta G, delta H, delta S. Then all these will be there in a QSR studies, molecular dynamic studies, quantum chemistry studies, and free energy calculations are the thermodynamical properties. All these we have to evaluate in terms of the computational. Then, so you use the graph neural networks or recurrent neural RNNs, then RCNNs will be there. So these all the machine learning models will predict the chemical properties and biological activity and drug target interactions, forming a core framework in a multi-layer. So I have a molecule and I'm representing the molecule in terms of my graph. So a number of the molecules I may take, I will have a network. That network will predict a molecular properties. So let us assume these all are the inputs. I have a 4 inputs, but when I read all these inputs properties, I may give you only one output. That is taking this sum, taking the weighted average, and I'm giving a sum. That sum will give a better function. That is trying to find out a better feature from the given representation of the molecules. Okay, here the graph theory, so node and edge is there. So node has a one property, edge has a one property. Then when you are combining, then the subsequent responsible prediction will come out. So that will be it. Okay. So we are trying to predict the property, and we are trying to identify in molecular generations, and we are trying to identify in descriptors. Then you have a generative models, transformer models, and GNNs. Generative models, the de novo drug design or exploration of a chemical space will be there, and multi-model optimization methods are. Instead of taking the one receptor, we also take the multiple receptors and we will perform the prediction studies, whether it is working on a liver or kidney or intestine or brain, like that. Multi-model optimization, multi-model, the modeling, multi-model studies also will be there in AI predictions. So then we have a transformer predicting models. So it is based on the property predictions, and that may be a sequence based, or that may be a transformer learning of a molecules. Then this is only network based approaches where the interactions are taking place. Then the AI and regulatory translation in the drug discovery. So these models will work. The explainable AIs, where the explainable AIs will give you the better compliance results, and it also accelerates the better development of a drugs with the proper the interventions. Then you have a data transparency. Whatever the data which you have, the data will be a reproducible. The data will have a proper transparency. There is no hidden data, there is no hidden results. Then validation and oversight will be there. So whatever the data which has generated, that data you need to validate, and you need to look at it properly, and you need to validate and give a proper results. So the classical approach to the modern AI techniques. So we have a transformation of the drug discovery with the AI interventions. The trial and error methods will be there, and cross validation and leave one approach will be there. So where you have a classical QSR models, only linear models, regression models, and correlations and regressions will be there, and that are the simple descriptors will be studied. But when it comes to the artificial intelligence and next generation AI models, we used to perform the machine learning studies and deep learning studies and graph models. And these machine learning and deep learning and graph neural networks models will give you the accurate predictions of your molecules by integrating with the large data sets. When you have a high volume of the data, that data may available in your electronic health records. That data may be available in your omics and real world data. So that data need to be represented in terms of a safer, in terms of a faster performance, in terms of a faster analysis, in terms of a target specific and therapeutic studies. So these days, one drug is made up for a one purpose, but when you goes into the population or polymorphism, the disease is one, but drug is not. So let us assume 100 patients suffering with some X disease. Only 20 members are cured with that drug. Remaining 80 are not cured. And out of 80, without taking any medication, 30 patients are cured, and remaining 50 are not cured. Then you have to give the combination therapy with the other drugs. Instead of giving a X drug, you have to combine that X drug with the Y drug, or Y drug with the Z drugs. So later they may be cured. So these days, all the medications are coming into a personalized medications or personalized drug design. Yeah. So these are some of the ADMET property tools like ADMET Lab and PSK, CSM and Swiss ADME, and there are all the tools that are available in your open source. You can just try in your web. And these are some of the AI-enabled tools like Topcast and Taskcast and PubChem and Chembel and DrugPen. So these are the AI models. These are also open source. This the AI enabled the drug and molecular discovery by using the computational platforms. So whatever the data which you are collecting, so various repositories are available today. So you no need to worry to synthesize the molecules. Lakhs of the molecules, more than 20 lakhs molecules that are there. Out of the 20 lakhs molecules, almost greater than 10 lakhs approved conditions are there. So that data need to be collected in terms of a chemical, in terms of the biological information, in terms of clinical information. Then whatever the data which you are collecting, then you have to pre-process. So the pre-process involves, first you have to clean the data. Once you have cleaned, based on your interest, based on your need, based on your study, then you have to normalize it. Once you normalize, then what is the target? The target model. So that AI models will analyze the data to identify a particular druggable target. So then you perform the virtual screening studies by taking the libraries, then screen it. Once you screen the large libraries data, then identify a lead molecules or do the molecular optimization studies or energy minimization studies by using the artificial intelligence and get the particular lead molecules. That lead molecule, optimized lead molecules, will conduct the in vitro and in vivo studies. Then that may be sent to the drug and marketing studies. Then, so these are some of the softwares and models, the ligand complexes, the FLAP and Farmer and ligand scout and catalyst and MOE and phase and other studies are there. So this will help us to understand. So this traditional QSR, so this is whatever the parametric models which we have, then this will be human engineered features will be there, and data driven and data limited sources that are there, and weak generalization properties are there. So when this traditional data, when you are conceptualizing into electronic form, then this complexity will be removed and the data scalability will be analyzed and intelligence will be extracted from the data sets. So once it is done, then it is easy to flexible modeling of the data, automating the data, then so data compatible will be analyzed. So when you have a machine learning based QSR, so here which we first study the physicochemical properties of a particular molecules, and you have a data acquisition and pre-processing of the scenario. And once you pre-process the data, you are trying to extract the feature. So once you extract the feature from the data, if anything goes wrong, again you re-engineering the data, then you remodel the data, changing the training set and test set values, then verify the data and evaluate again and engineering the identification of the prioritization of the data. So once it is done, the prioritizing of the data, then we will extract the feature. So the large, today the BERT and ChemBERT and LLMs are there. So these are the large language models that are giving the better results, where you have a conformational predictions and physics informed physics networks and neural networks, and again agentive AI models or generative AI models and explainable AI models. These all will predict the technological innovations, the strategic responses, and other things. So like this, the studies will be there, and so let's skip. Yes. So these are the data sets normally which we used to use. So GDSC database is there, and so normally we know when you wanted to do the cell culture studies, only 2-3 cell lines we used to say. But for the breast cancer, we have computationally 52 cell lines are there. Computationally available cell lines 52. And we have taken the anchor drugs is 25 and the library drugs are 21, and different pathways and number of the combinations and number of the entries. And we need to search a number of the databases to collect the data. then you need to do the model studies and model passports, and you need to see the molecular descriptors for the calculations of a molecules. So topological descriptor, geometric descriptor, electronic descriptor, physiochemical descriptors, three-dimensional descriptors, and molecular complexity descriptors. So like this, whenever you collected the data, all these properties you need to check in terms of your CSV file, in terms of your Excel file. Then, so this we have studied about the QSR, and so here we are collecting the data. Once it is collected the data from the GDSC databases and pre-processing the data, and based on the anchor docs and based on the library preparation in the based on the molecular descriptors, then we have exploratory data analysis were conducted by using the machine learning and deep learning architecture, and we have got a better results. So this is a one way, and this is a second way which we perform. So this is exploratory data analysis by using the GTSC multi-data set combinations. Then, so we used to do the different types of the heat map generations and other things, which this is already all this data is published in, and it is available in various publications. And these are the validation matrices. Validation matrices we used to follow by root mean square error analysis and mean absolute error and AOC curve and RUC curve and R square value and Q square value analysis to be conducted. And this another objective is there. So this is the regression plots. So look at here, initially when you are pre-processing, before pre-processing, after pre-processing, and after normalizing and making a cluster, and analyzing the particular regression values into grouping into particular properties. And then these are the different MDMB properties. And this is another set of the molecules, and where you are combining the molecules with the 6 combinations. And we screen more than 10 lakhs molecules, but when we have a different combination, the top 6 combinations which we made, and these molecules were shown here better results to against the breast cancer pathway, breast cancer receptor, as well as the EGFR pathways. And this is the sharp analysis, and we'll skip due to the time constraint. So this is I wanted to show one is another approach. So this is multi-model using the deep neural networks. In between, if you have any questions, you feel free to ask me. To the time constraints, I'm skipping slides. Just I want to show one representation. Let's close it. This is a multi-model. See here, we have generated a resistance network that is called a hybrid neural network model for predicting the drug sensitivity as well as the drug repurposing for a breast cancer. So we are working for a various cancers in the repurposing of a drugs instead of developing already existed drugs which we used to use, and we will study for some other purpose. That is called resistance. So this resistance here, so we have a molecular sequence, receptor sequence, the target protein or target transcription factor or target enzyme or target gene sequence which we take. Then we will take the drugs which are available in the libraries. Then we will study the descriptors. Then once you study the descriptors, and we will take the patient's data, then we will, whether the patient data is a malignant or benign, then we will conduct the machine learning. We will generate a machine learning models and conduct the neural network studies and deep neural network studies. Then one convolutional neural networks, one-dimensional, two-dimensional, and three-dimensional convolutional neural networks we will study. And when you take all these type of the data sets, our networks will be able to predict the sensitivity and specificity of the molecule. So when you have given a particular target sequence of a receptor and you have a drug and combination of a drug or anchor drugs, then you have a pathway. Then you have a patient. Whether this will works against the breast, against the liver, against the lung, against the other target. So based on the molecular sensitivity, specificity, based on the target regions, so our model predicts the particular property. So that is called the resistance network model. So this resistance network model will predict well, which were published in scientific reports. So evaluates the accuracy, recall function, F1 score, and precision, and MCC. So these are the completely statistical parameters in terms of the machine learning and AI ML studies. So these studies will give you better results, and this is what we will publish. So with this, I will stop it here. If you have any questions, you feel free to ask. Slides are there.
Participants have any questions, please free to ask. Participants have any doubt?
Sir, you speak.
Yes, sir. Please go ahead, sir.
Sir, are there any free tools for pharmacophore modeling?
So there are many free tools are available, sir. So whether you wanted to, you have a molecules or you wanted to search the database.
Sir, for generating pharmacophore models.
Yes, you can generate a model, sir, but basic scaffold you require, no?
Okay, sir.
So basic the scaffold based you can predict, or you can use some of the side chains and R1, R2, R3 groups. Sir, I'm closing the session. I'm coming.
Let's say if we have already a pharmacophore, for example, we have a pyrrozole ring. So then, what are the tools we can use?
Madam, you have a paddle and you have a PubChem. So PubChem itself you can search, and Malinspron is there, and this ACS softwares models are there. Even today you have this. There is a good software that is called molecular solution software, AI enabled feature. So this is predicting like anything. So trillions of the molecule it search and it will give you a proper molecule for your studies. Molecular solutions. It is a new software which is developed with the AI, but it is an expensive software. And you can search the Sibyl, Tripos, Sibyl, or Schrodinger, and Biovia. So they have a limited parameters to search the databases. But there are a number of the databases are available. So you pharmacophore database search just you can give, and it will give you better results.
Okay, thank you.
Good afternoon, sir. That was a wonderful presentation. I am glad to hear from you regarding the QSR aspects and connected with the ML, artificial intelligence, etc. Sir, would you let us know any? Can you suggest any free tools which can be used for this?
Madam, PharmaDB.
Mm.
PharmaDB, that means pharma database. Then farm mapper.
Farm?
Farm mapper. That is pharmaceutical mapper.
Uh-huh.
Farm target DB.
Okay.
Farm target DB. Pharma DB. Farm mapper. ADME mapper, etc., etc. There are the many, madam. Just I'm giving a few.
Are the means I would like to know, will the data be reliable when we go with the
Reliable, madam.
versions
Reliable. All are the
when
public
compared with the
sources, public
subscription versions?
repositories. They are reliable only, madam.
Yeah.
We are working with the public data only.
But there was a doubt regarding when we go with the online versions, can we rely on the data, or is it a must to go with the subscriptions to certain extent? But
No, that is a human tendency. First, initially, they will habituate to the addict to that system. Once you are addicted, then you have to go for a subscription. So we don't comment anyone.
Okay, sir. Understood. Thank you.
Okay.
Good afternoon, sir.
Good afternoon.
Yeah, I'm Vinuta, sir. Sir, previously we have tried on the free software like Pharmagist, which was operated for pharmacophore modeling. Nowadays we are unable to access that site, sir.
Because earlier it has a freeware, now most of the softwares are commercialized now. Because it's earlier they tried to addict to that software, and you have a good command in that software. Once it is given here for a open source for a couple of years, now they were converted that open source into a commercial. Then you don't have an option to access that.
Is there any free available software like that, sir?
I can't say one, madam, but there are many. Just you search, madam.
Okay, sir. Thank you.
Yeah, so any other questions, sir? If no more questions, sir, we thank you, sir. So as you know, like pharmacophore plays very important role in drug discovery, and it serves as an foundational blueprint in drug discovery to identify, filter, and improve potential candidates. And AI-enabled drug discovery is a recent like norms what we are seeing. And so we thank you, Dr. Perigu Shyam sir, like for providing an elaborate understanding on the pharmacophore modeling and AI-enabled pharmacophore modeling, and like the basics, complete basics, and also what are the advanced AI techniques, how we can use in pharmacophore modeling. And on the behalf of JSS Academy of Higher Education and Research, JSS College of Pharmacy, UT Department of Pharmaceutical Chemistry, we thank you very much for providing your time and for explaining our participants these new techniques. Thank you, sir.
Thank you, Ananda, for giving the opportunity. If you have any queries, and you email to me, or if any work to be work with the collaboration, or any docking studies, dynamics, and any AIML studies, feel free to write to me, and I will help you. Thank you. Thank you,
sir.
Ananda.
Yeah. So we can conclude the session. Thank you all participants for joining the session and active listening, for your active listening.
Sir, can you please give us his mail ID?
Yeah, we will provide the details. Yeah.
Thank you, sir.
We'll end the session.